Hello everyone and welcome to K Foundations. Myself Uthara Adithi, and today we'll be continuing with the chapter Cell the Unit of Life. So we were supposed to do these diagrams, right? Yeah. So firstly, here's your rough endoplasmic reticulum. You can differentiate it. Here you can find the smooth endoplasmic reticulum. The one with these structures on top is your rough endoplasmic reticulum, right? And that's the nucleus. There's the nucleolus. Here we have our Golgi. There's the nuclear envelope, plasma membrane. There's a large vacuole, right? And there's this middle lamella, there's the cell wall, mitochondria. If you note, ribosomes, peroxisomes, and lysosomes, they're all spherical, so you have to see how they are different by their size, okay? So next is our chloroplast, cytoplasm is there. Then we have microtubules, plasmodesmata, smooth endoplasmic reticulum, right, without any ribosomes. There's the lysosomes. So yeah, next over here, an animal cell. They're possessing microvilli, plasma membrane. There's no cell wall, right? Yeah, these have centrioles because animal cells have centrioles while the plant cells don't have centrioles. Here there are peroxisomes, lysosomes, ribosomes, mitochondria, single mitochondrion, rough endoplasmic reticulum, cytoplasma, and then there's the nucleus, nucleolus, in between, nuclear envelope, smooth endoplasmic reticulum here, and Golgi apparatus. Okay, there. Now let's go to the cell membrane part. Yeah. The detailed structure of the membrane was studied only after the advent of an electron microscope, okay, in the year of 1950s. Okay, in the during the years of 1950s, electron microscope was found and was prepared, and then through that we were able to see cell membrane. Meanwhile, the chemical studies on the cell membrane, especially in the human RBCs, enabled to deduce the possible structures of plasma membrane. We were able to study well about the plasma membrane while studying the human RBCs. Okay, well, later when the electron microscope was found, it became much more easier. These studies show that the cell membrane is composed of lipids and proteins. Okay, so the major lipids are phospholipids, and that's why it's also called as a phospholipid bilayer. Okay, as per this diagram we'll understand that. So the cell membrane has lipids and proteins. The major lipids that are found are phospholipids. It's arranged in a bilayer. Also, the lipids are arranged within the membrane with the polar head towards the outer side. If you see towards the outer side, there's this polar head, and there's a tail as such, which is a non-polar tail. You can see two tails here actually. Okay, you can see that way. Yeah, and the hydrophobic towards the inner side. Okay, hydrophobic tails. So this is the hydrophilic polar head, and here you'll find the hydrophobic non-polar tail. Okay, just the opposite. This ensures that the non-polar tail of the saturated hydrocarbon, which type of hydrocarbon? It's a saturated hydrocarbon, is protected from the aqueous environment. Okay, what is hydrophilic and hydrophobic? Hydro is water, philic means love, phobic is to hate. Okay, so water loving components are towards the outside, and the water hating, or the ones that are phobic towards water, are towards the inner side. Okay, so in addition to the phospholipids, they also contain cholesterol. So in terms of lipids, they contain phospholipids along with cholesterol. Later, biochemical investigations clearly reveal that the cell membrane also possesses proteins and carbohydrates. This ratio between the proteins and carbohydrates can vary up to different cell type to cell type. It will vary. Okay. In human beings, the membrane of the erythrocyte RBCs are approximately 52% proteins and 40% of lipids. Okay, this is 52% of proteins, more amount of proteins than the lipids. Okay, so now depending on the ease of extraction, what is a criterion for classification of these membrane proteins? It is based on the ease of extraction. They are divided into integral and peripheral proteins. Peripheral proteins, periphery, to line the periphery on the surface. Okay, while the integral proteins are partially or totally buried in the membrane. Integral is towards the inner side, I mean it's buried partially or wholly. Okay, so this is the structure. If you see the hydrophilic heads are towards the outer side, while the hydrophobic tails, here are the tails which are found towards the inner side. Let's see it. Right, hydrophobic tails are towards the inner side. So now, yeah, if there's water outside, okay, here's water and there's water here, here is also, there's also water. Okay, so this the outer side which likes water will not be phobic towards it, right? But if the reverse was made, let's suppose the layer was like this and there's water outside, then these are hydrophobic, right? They don't want water, so their functions will necessarily not be taking place. Okay, okay, yeah. So there's this sugar, there's the peripheral protein. If you see, this is the peripheral protein found on the periphery. Okay, and this is the integral protein. Here it's not totally buried, it's like partially buried. And there's this phospholipid bilayer, and here this yellow kind structures is called cholesterol. Okay, so cholesterol. This is the fluid mosaic model of plasma membrane, which is given by Singer and Nicholson in the year of 1972. We'll be seeing that. Okay, yeah. An improved model of the structure of cell membrane was proposed by Singer and Nicholson. I mean, many models were proposed previously, but then the accepted ones are by Singer and Nicholson in the year of 1972. It is also called as the fluid mosaic model. Okay, what does this model say? There's this quasi-fluid nature of the lipids which enables the lateral movements of the proteins within the overall bilayer. There's this bilayer, right? So the movement of proteins within the bilayer was possible because of the fluid nature of lipids. This ability to move within the membrane is termed as fluidity. Okay, the ability to move is termed as fluidity. Now, what is the role of this fluid nature? Why does the membrane possess it? It is having certain functions, like it helps in cell division, cell growth. Cell division will be also be there. Cell growth, formation of intercellular junctions, secretion, endocytosis, and cell division. These are the roles. What are the 5 roles? You can remember cell growth and cell division at once. Secretions, formation of intercellular junctions, and endocytosis. Okay, so these are the functions of the fluid nature of the membrane. Okay, one of the most important functions of the plasma membrane is the transport of molecules across it, of course, right? It's in the outermost layer, so it does some work in enabling the transport of substances either from outside to inside or the reverse. The membrane is selectively permeable. It doesn't allow every kind of substances inside, right? Yeah, so it's selectively permeable to some molecules present on either side of it. Many molecules can move briefly across this membrane without any requirement of energy. That is called as a passive transport. There are two types of transports, passive and active. In passive, there's no necessity of any energy. Active requires energy. The neutral solutes, they are neutral. They don't possess any charge. They're not, a polar or non-polar. Okay, they are neutral. Yeah. So may move across the membrane by the process of simple diffusion. It's neutral, right? So it can pass through the membrane through the process of simple diffusion along the concentration gradient to be noted. It's like from the higher concentration gradient to the lower. That's why it's called as a passive transport. If reverse needs to be done, you'll get to know that it's an active transport. Water. Movement of water, I'm pretty sure you'll be knowing it. From high to low concentration is termed as osmosis. Okay, this is also by diffusion, a special type of diffusion called as osmosis. As the polar molecules cannot pass through the non-polar lipid bilayer. See, towards the outer side there's polar, okay, fine, but then towards the inner side you'll find the non-polar tail. If the polar molecules have to be transported, then they'll come in contact with the non-polar lipid layer, but then they don't want to do that. So what happens is we use certain carrier proteins, okay, carrier proteins of the membrane to facilitate their transport across the membrane. A few ions or molecules are transported across the membrane against, yeah, when you're transporting certain molecules against the concentration gradient, that is called as an active process. It's an energy dependent process where what is the energy? ATP. ATP is being used. Okay. Now, Na plus K plus pump is an example of that. Okay, so we came across different types of transport, passive and active. Under passive, we saw simple diffusion, again osmosis, and then we also saw with the help of carrier proteins, right, facilitated diffusion. And active diffusion, what was the example? Na plus K plus pump. Okay, now we'll be seeing about the cell wall. Yeah, as you may recall, a non-living rigid structure to be noted, it's a non-living structure. We know that it's rigid structure, right? That's why it's protecting the cell, the cell, and it has many roles. So it's a non-living rigid structure, forms an outer covering for the plasma membrane of fungi and plants, right? Apart from animals, most of the other organisms possess cell wall. But then there are exceptions, like you won't find them in mycoplasma, okay, which is a monarin. Now, cell wall not only gives the shape to the cell, it does give shape to cell, but also it protects the cell from mechanical damage and infection. Any kind of damage, it will protect it. Infection, it won't allow them. Okay, it also helps in cell to cell interaction. Okay, adjacent cells, if they have to interact for the transport of certain molecules, then the cell wall plays an important role in that. Okay, and it provides a barrier to undesirable macromolecules. If any kind of macromolecules which are undesirable, which is not necessary, or it could be, it could cause any damage to the cell, then cell wall will prevent that. Okay, so algae have cell wall made up of cellulose, galactans, mannans, and minerals like calcium carbonate. Cellulose, galactans, mannans, minerals like calcium carbonate. While in other plants you can find cellulose, hemicellulose, pectins, and proteins. Okay, algae and on the other hand, plants. Algae, to be noted, both of them have cellulose, cellulose, cellulose. Okay, so in plants, as a continuation, hemicellulose, pectins, and proteins. On algae side, we can find cellulose, galactans, mannans, and minerals like calcium carbonate. The cell wall of a young plant cell. Speaking about a young plant cell, there are different layers. There's a primary cell wall which is capable of growth, okay, and once it matures, it which gradually diminishes as it matures, a secondary cell wall is formed on the inner towards the membrane side of the cell. Here, let's suppose that this was the primary cell wall. Okay, so now what happens is it grew. It, of course, it should be able to grow, right? It grew, and then now it became thin. I mean, it differentiated, it became matured. Okay, it became matured. This primary cell wall became matured. Now the secondary cell wall will be formed, right? That will be formed inner. Okay, it's formed on the inner side. Let's suppose here's the nucleus and all of that. So towards the inner side you can find the secondary cell wall. Okay, yeah. So the middle lamella, there's a middle lamella, okay, will be on the outer side here. Here you can find a middle lamellae. Okay, middle lamella is a layer of calcium pectate. It also contains magnesium pectate, okay, which holds or glues the different, yeah, the role of it is to hold or to glue the different neighboring cells together. Adjacent cells, they have to be together, right? It isn't like the cells are far apart. They should be held together, and that gluing function is done by middle lamellae. Okay, the cell wall in the middle lamellae may be traversed by plasmodesmata, which connects the cytoplasm of neighboring cells. Yeah. See, let's suppose this is the cell, right? So there are connections like this. So again, there'll be this middle lamella of the adjacent cell, and then there'll be primary cell wall, secondary cell wall, right? So there'll be connections like this. Okay, so through these connections, sometimes smaller particles will travel. Okay, sometimes huge ones traveling also it will allow, like this. So connection of the cytoplasm, here also there's cytoplasm, right? Cytoplasm number one, and there's cytoplasm number two. It helps in connection of that. Okay, now we'll move to the endomembranous system. Okay, while each of the membranous organ layers is distinct in terms of its structure and functions, many of these are considered together as endomembranous systems because their functions are coordinated. The important reason for this endomembranous system, it comprises first of all of four parts. Okay, your ER, GC, and lysosomes along with vacuole. Why are these four specifically called as the endomembranous system? Is because their functions are coordinated. The endomembrane system includes the endoplasmic reticulum, endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. We'll be studying about them as well. Okay, since the functions of the mitochondria, why not the rest of the mitochondria, chloroplast, peroxisomes? We have other organelles too, but they aren't a part of this because their functions are not coordinated. Simple reason. Either your functions are coordinated, become a part of the endomembranous system. If it's not coordinated, it will be not a part of it. It won't be a component of it. Okay. Now, the endoplasmic reticulum (ER), the electron microscopic studies of the eukaryotic cell. Okay, reveal the presence of a network of reticulum of tiny tubular structures scattered in the cytoplasm called as the endoplasmic reticulum. Okay, so this electron microscope has helped a lot. So two questions. First of all, around which year was it found? And also, it helped in it helped in studying which part of the cell much better. The cell membrane, yeah. And then in which year? It was around 1950s, right? Yeah. Hmm. So now it's coming back to endoplasmic reticulum. It's a network or reticulum. If you see this, it's a network or a reticulum. It's not properly arranged. It's kind of like this, right? Uh-huh. So it's scattered in the cytoplasm, and this is called as endoplasmic reticulum. This endoplasmic reticulum divides the intracellular space into two distinct components. The intracellular space, okay, within the cell, it is dividing the cell within it into two parts, the luminal and the extraluminal part. The luminal is inside here and the extraluminal is in the cytoplasm here. Luminal, extraluminal, all of these. Okay. So ER often shows ribosomes attached to their surface. Okay, so the ones that have ribosomes on their surface will become rough, and it's called as rough endoplasmic reticulum or ER. Okay, in the absence of ribosomes, they'll be smooth, they'll appear smooth. This is called as the smooth endoplasmic reticulum. Okay, rough endoplasmic reticulum is frequently observed in the cells actively involved in protein synthesis. Yes, and secretion. Rough endoplasmic reticulum possess ribosomes. Those ribosomes involve actively in protein synthesis and secretion. They're extensive and they're continuous with the outer membrane of nucleus. While studying nucleus also will know that rough endoplasmic reticulum is in continuity with the outer membrane of the nucleus. Nucleus is having two membranes. In that outer membrane is continuous with rough endoplasmic reticulum. Smooth endoplasmic reticulum is a major site for synthesizing lipids. Okay, steroidal hormones and all you can synthesize. In animal cells, lipid-like steroidal hormones are synthesized by SER, smooth endoplasmic reticulum. So in this diagram, we can see the nucleus. There's the nuclear pore right here. And if you see the outer membrane, it's continuing from the outer membrane. That's why this rough endoplasmic reticulum is continuous with it. You can find ribosomes on their surface right here. That's why it's called as rough endoplasmic reticulum. Ribosomes, smooth endoplasmic reticulum. Okay, moving to Golgi apparatus. Right. Camilo Golgi, there of 1898, firstly observed this densely stained reticular structure near the nucleus. Okay, it's a densely stained reticular structure. These were later named as Golgi bodies after him. They consist of many flat disc-shaped sacs or cisternae. You can remember that Golgi had a sister, so it's called a cisternae. These are called as cisternaes of 0.5 to 1 micrometers in diameter. Important. Okay, 0.5 to 1 micrometers in diameter. You can remember that 0.5 times 2 gives you 1. So 0.5 to 2, 0.5 to 1 micrometers diameter. Okay, yeah. So these are stacked parallel to each other. Varied number of cisternae are present in the Golgi complex. It's not fixed. The Golgi cisternae are concentrically arranged near the nucleus. Okay, they're arranged near the nucleus with the distinct convex cis or the forming phase. They have two. These have two phases. There's the cis convex or the forming phase, and the concave trans or the maturing phase. Okay, the cis and the trans phases of the organelle are entirely different. The cis and the trans are entirely different. That's why one is forming, one is maturing, one is convex, the other is concave, but they're interconnected. You can find certain interconnections between them. Okay, the Golgi apparatus will be seeing the role of it principally performs the function of packaging materials to be delivered either to the intracellular targets or secreted out of the cell. So just like our endoplasmic reticulum, we saw rough endoplasmic and smooth endoplasmic reticulum. And under rough, what were their roles? It helped in protein synthesis and secretion, while in smooth endoplasmic reticulum, it was helping in steroidal hormones formation, lipid synthesis, and all. Now, Golgi complex, here it helps in packaging of those materials. Okay, and either you can, either those are supposed to be secreted out of the cell or to be utilized within the cell. Materials to be packaged are in the form of vesicles that move from ER and fuse with the cis phase of the Golgi apparatus, and they move towards the maturing phase or the trans phase. Okay, this explains why Golgi apparatus remains in close association with endoplasmic reticulum. Okay, because ER, whatever the products are there, they need to be packaged and then they need to be transported. Both the works Golgi does. Golgi helps in packaging them. They form the substances into vesicles. You know they'll fuse with the Golgi as vesicles and then removed. And yeah, okay. A large number of proteins synthesized by ribosomes on the endoplasmic reticulum are modified in the cisternae of the Golgi. Modifications in the cisternae before they are released from the trans phase. Golgi apparatus is the important site for the formation of glycoproteins and glycolipids. Okay, Golgi apparatus role is also to synthesize glycoproteins and glycolipids. So first role is for packaging, right? Another is for the synthesis of glycoproteins and glycolipids. Moving to lysosome, the third part of our endomembranous system. These are membrane bound. Okay, no doubt about that. The membrane bound vesicular structures formed as a process of packaging in the Golgi apparatus. So, one such organelle is itself formed as a result of packaging in Golgi apparatus, which is lysosomes. The isolated lysosomal vesicles have found to be very rich in almost all type of hydrolytic enzymes. Okay, these hydrolytic enzymes, it includes like lipases, proteases, carbohydrases, optimally working at an acidic pH. It's like helps in digestion kind. Okay, the isolated ones have very rich hydrolytic enzymes which work in an acidic pH in order to break down, digest carbohydrates, proteins, lipids, and nucleic acids. Like lipases will be for lipids, proteases for proteins, likely. Okay, yeah. Last part, vacuoles. Yeah, the vacuole is a membrane-bound space. So up to now, we didn't come across an organelle which is not membranous in ER. In the endomembranous system found in the cytoplasm. It contains water, sap, excretory products, and other materials which are not useful for the cell. The vacuole is bound by a single membrane called as the tonoplast. Tonoplast is called as the basically the membrane of vacuole is tonoplast. In plant cells, the vacuoles can occupy up to 90% of the volume of the cell in plant cells. I said this previously as itself. 90% of the plant cell, the volume of it is occupied by the vacuoles. In plants, the Tonoplast facilitates the transport of a number of ions and other materials against the concentration gradient into vacuoles. Means this water, sap, excretory products, when they are just accumulated in the vacuoles, there'll be more in concentration in the vacuoles rather than in the cytoplasm. So it should be pushed against the concentration gradient. Hence, their concentration is significantly higher in the vacuole than in the cytoplasm. That's why, right? It requires a certain push. Yeah. In amoeba, the contractile vacuole, okay, in amoeba, you can see the contractile vacuole which helps in osmoregulation and excretion. While in many cells, as in protists, food vacuoles are formed for engulfing the food particles, right? So we have our amoeba. This is amoeba as well as our protists. Okay. Amoeba has contractile vacuole while protists have food vacuole. Okay. Contractile vacuole helps in osmoregulation and excretion, correct? And food vacuole helps in engulfing the food particles. I mean, food vacuole engulfing the food particles makes sense, right? Yeah. So now about the mitochondria and our plastids, and further information will be done in the next video. Until then, definitely spread the key of success, spread knowledge. Thank you.